PG-30, The Key Study Guide, Alberta Edition, 2015. Ataxia telangiectasia, AT, is an autosomal recessive disorder occurring with an estimated frequency of 1 in 40,000 births. The first symptom of the disease occur around 2 years of age and are progressive. They consist of a lack of balance and a slurred speech. Soon after, tiny red spider veins appear in the corners of the eyes, telangiectasis. Older children with AT lose their ability to write and reading becomes impossible as eye movements become difficult to control. Individuals are eventually confined to a wheelchair. Immune system disorders are common. Intelligence is normal in these individuals. Most individuals with AT die as children. However, some live up to 50 years. Patients with AT develop blood system cancers 1,000 times more frequently than the general population. Treatment of cancer with conventional dosage of radiation can be fatal to AT patients because they are especially sensitive to radiation, which causes breakage of DNA. Even carriers have a higher incidence of cancer than does the general population. The defect causing AT has been traced to a mutation on chromosome 11. The protein product of this gene is not expressed appropriately. The exact role of the gene remains a mystery. Normal cells respond to DNA damage by interrupting the cell cycle in interphase before DNA synthesis occurs. This allows for repair of the DNA. Scientists are investigating a link between the AT mutation and this cell repair process. There is presently no cure for AT, but treatments to alleviate symptoms are currently being developed. On April 26, 1986, one of the worst technological, industrial, and environmental disasters known to humankind occurred. A nuclear reactor in Chernobyl exploded and showered radioactive debris over much of Eastern Europe. The extent of the environmental and health effect of the nuclear legacy of the Chernobyl disaster are still unknown. Although the exact cause of many illnesses are not understood, there is little doubt that the enormous burst of radiation released from the reactor has had a devastating effect on the thousands of children. One of the most dangerous radioactive product released was iodine-131. It was inhaled by many children, exposing them to high levels of radiation. Iodine-131 was absorbed by the children's thyroid glands, causing inflammation of the gland and an increased inflammation of the gland incidence of thyroid cancer. Normally, iodine is absorbed from the blood by the thyroid gland in its synthesis of thyroxine. The effect of this radiation over a long period of time were also studied. Researchers looked at DNA gene sequence 5 to 45 base long from some blood samples taken from parents and other children born in 1994 or later. They looked for any sequence in the child's DNA that did not occur in the blood cells of either parent. The children born near Chernobyl had twice as many of these mutations in their DNA as had the control group, which consists of families in England whose children were also born in or after 1994. There is a worldwide shortage of organs for transplanting into humans. Some researchers are concentrating on xenotransplantation, using organs from other species as a solution. The pig is considered by researchers as the most suitable donor of transplant organs for humans, even though pigs are not as closely related genetically to humans as higher primates are. Pigs can be bred easily and up to 3 times a year. Sows have short pregnancies, about 15 days, and give birth to large litters. The offspring grow quickly to reach a large size. Transplant organs from ordinary pigs are quickly rejected by the human immune system. Researchers have isolated a human gene called HDAF that codes for a cell membrane protein known as RCA. The human HDAF gene can be inserted into pig DNA, so the human RCA protein will be present on the surface of pig cells. Human RCA protein on cell membranes of pig cells is expected to inhibit the rejection of harvested from a cell, injection of HDAF gene into the nuclei of the fertilized eggs, identification of heterozygote transgenic offspring, shade gray, expressing RCA protein in their organs, selected heterozygote transgenic pigs are crossed, identification of homozygote transgenic offspring, shaded dark gray, expressing RCA protein in their organs. Approximately 60% of these offspring were transgenic heterozygotes. Scientists are working hard to learn about cause of cancer. It is known that in general terms, cancer-causing agents produce mutations in a variety of genes that control cell reproduction. Cancer can be caused by organisms. For example, when some viruses infect a human cell, the viral DNA may insert itself randomly into the DNA of human cells. Of all the cancer-causing agents to which humans are commonly exposed, tobacco smoke appears to have caused the greatest harm. It is directly responsible for 30% of all cancer deaths in North America. Heavy smoking increased the likelihood of getting lung cancer by 2,000%. Passive secondhand smoke is less likely to cause cancer. It is about as harmful as all other forms of air pollution. 4% of all cancer death can be linked to the reproductive history of a woman. Researchers have discovered that if a woman begins to menstruate at an early age and experience late menopause, she is more likely to develop cancer in her reproductive organs. If a woman has several children and gave birth to them at a younger age, she is less likely to develop cancer of the ovary, breast, or endometrium. Scientists have been learning about a mechanism called the cell cycle clock that collects information from outside of the cell. This information influences the activity of molecules within the cell that determine whether or not a cell will undergo mitosis. Some molecules take the form of quote-unquote go signals, stimulating a cell to go through mitosis. Other molecules take the form of stop signals to ensure that the cell does not start cell division. For example, some cyclins and CDKs are molecules that act as quote-unquote go signals. If they are present in appropriate amounts, DNA replication will occur. Adrenoleukodystrophy, ALD, is a rare disease of the central nervous system. ALD is characterized by the accumulation of very long-chain fatty acids in the white matter of the brain and in the adrenal glands. These fatty acids cause the myelin sheath on nerve fibers within white matter of the central nervous system to degenerate. Symptoms of this degeneration become more severe as more and more fatty acids accumulate. Symptoms start with tantrums and other behavioral problems, then motor function, speech and hearing are impaired, and finally blindness, mental deterioration, and death occur. ALD also affects the endocrine system by causing adrenal gland degeneration. ALD can be partially diagnosed by abnormally high ACTH levels in the blood. Hereditary disease have diverse causes. For example, the disease mutation may be dominant or recessive, or the mutated gene may be present on the X chromosome or on an autosome. In some cases, similar disease are caused by mutations in two different genes. One such case is ALD, where one gene is autosomal and the other is X-linked. In both forms of inheritance, the disease mutation is recessive. Scientists continue to research the cause of ALD. The X-linked recessive form of ALD can be diagnosed prenatally. ALD has not been treated successfully. However, bone marrow transplant and a diet restricted in very long-chain fatty acids have shown promise. One dietary substance, oleic acid, Lorenzo's oil, has been successful in normalizing levels of very long-chain fatty acids in blood plasma. However, the results of clinical trials on ALD patients show little improvement in symptoms, particularly in brain degeneration. Lorenzo's oil did produce positive clinical results when the treatment of patients began before neurologic symptoms were present. Acquired immune deficiency syndrome, AIDS. Research has centered on developing drug treatment and an AIDS vaccine. AIDS is caused by human immunodeficiency virus HIV, the drug AZT can greatly reduce the chance of transmission of HIV from an infected woman to her unborn child. The current AZT treatment is very costly. To make the treatment more affordable, African researchers have conducted a number of studies in which one group of HIV-infected women was given a shorter than normal course of AZT treatments, and another group of HIV-infected women received a placebo pill without medication. A vaccine made from only the outer coating of the HIV is attached to a harmless virus. The vaccine is being tested on healthy human volunteers to see whether they develop antibodies that would help them produce a natural defense against AIDS. This type of vaccine has not yet been sufficiently effective to induce the desired immunity. A similar AIDS vaccine test in Canada did not work any better than a placebo. Future treatments for AIDS may be based on newly acquired knowledge of how AIDS infection occurs and why some individuals are more resistant to infection. When body cells are damaged, they produce a protein, chemokine, to attract the body's immune cells, macrophage. The macrophage have receptors on their cell membranes that attach to the chemokine and rid the body of the damaged cells. The HIV attaches to one kind of chemokine receptor, CCR5, on the macrophage and enters the macrophage, but the HIV does not destroy it. Ultimately, the virus also infects T cells and takes over their DNA replicating mechanisms. The macrophage infection may be necessary to activate the replicating mechanisms of T cells. It has been discovered that some people have inherited a resistance to HIV because the gene that makes the CCR5 receptor is mutated, missing 32 nucleotides. This CCR5 mutation results in a shorter receptor, thereby preventing the HIV from attaching to macrophage. Individuals in a study group were homozygous for the mutant allele-resistant infected despite many exposures to HIV. Individuals in the study group with one copy of the mutant allele had the onset of AIDS postponed for 2 to 3 years when compared with those in the study group that had no copies of the mutant allele. The mutated allele is most common in Caucasians. Human growth hormone HGH stimulates the growth of bones and muscles and also has insulin-like properties that result in the deposition of fat and body tissue. Sometimes, for medical purposes, an individual is prescribed a supplement of HGH. Unfortunately, at high levels, HGH has some unpleasant side effect. At a high dosage, HGH can cause insensitivity to insulin. In a extreme case, permanent diabetes mellitus can result. Dr. J.F. Mueller made an interesting discovery. He observed that mice infected with the Pleurocercoid larvae of the tapeworm Spiometra mansonoides showed accelerated growth. Careful analysis revealed that the larvae produced Pleurocercoid growth factor PGAF that is remarkably similar to HGH in structure and function. Further studies showed PGF to be molecularly similar to HGH, but it had no anti-insulin effect. How is it that a tapeworm larva produces a substance so similar to HGH? Even closely related animals have quite different hormones, so it is not likely that this is a chance occurrence. One hypothesis that has been suggested involves viral transduction of the HGH gene. When a virus infects a host cell, the viral DNA becomes incorporated into the host DNA. Later, the host cell is tricked into assembling new viruses. The viral DNA is replicated and placed in the new virus. Sometimes a section of host DNA is added along with the viral DNA. If the new virus infects a second host, the second host receives the DNA from the first host. This host-to-host transfer of DNA is called viral transduction. The life cycle of the tapeworm allows it to easily infect not only humans but several other vertebrates. Almost Almost all tapeworms are hermaphrodites, contain both male and female sex organs. Multiple testes and ovaries found in segments on a single tapeworm produce gametes that come together and result in the production of fertilized eggs, neither by self-fertilization or cross-fertilization.